All right, so I'll try to run through this a bit quicker for you guys. So Amplicon Metagenomic Sequencing. Essentially, the same thing that was just talked about, but we're doing a PCR product that was mentioned in the back earlier, right? So what 16S, 18S, and ITS. So we have bacterial, here we are. Bacterial, eukaryotic, and fungal rRNA components that, so primer sets have been established for them. rRNA is highly conserved across these individuals. And so what we can do, because of the conservation across the individuals, we can look at subtle changes that may lead to a community profile change. And so this is one way to monitor a community or monitor a divergence in an isolate. So they target the gene specifically. They survey microbial samples for diversity and helps identify the species in the sample. The generalized workflow is here. So we're going to be taking our raw sample, processing it in some way. At Novogene, we do this end-to-end, so you just give us the DNA. But we'll use primer sets, or if you give us primer sets, we can generate those. And we push all the way through extraction, library purification, quantification and pooling, and then sequencing. One thing I do want to mention is third-generation sequencing here. So this is an option for the same reasons that Kyle brought up earlier. Using a combination of Illumina and PacBio can help you better understand your organism. Caveats would be cost, redundancy, size of data needed, but it is something that is offered and it is something that is doable. So just again, for you guys' reference, some ideal extraction methods for metagenomics. In general, in my opinion, you don't have to be as neat with DNA as you do RNA. So these extractions can be a bit messy, if you will. Like you can put a lot of stuff into these columns and you'll still get community DNA. Whereas you may need to be salinity controlled, temperature controlled for specific RNA from different environments to elute out of a column. So these are some examples of known extractions. CTAB, magnetic beads, pretty common. So I want to show you guys the Amplicon Metagenomic Sequencing analysis pipeline. So Kyle just went over one previously. Very simple. Raw data. I guess I should do this. Raw data at each pipeline that Novogene offers, we do data quality control before the client even gets the data. This ensures our viability of our pipelines. Cleaning the data up, OTU clustering. So we either do alpha and beta. You can do either one or the other or both or combination. You're the client. So alpha diversity measures the diversity within the sample group. Within the single sample. Beta diversity measures the difference between samples. So both alpha and beta diversity are important in understanding the structure of your microbial communities. So within and across samples, and then just as the other pipeline, you can do enrichment analyses, lineage analysis, and this would all be off of a PCR product. So comparison here, which one should you do? So this paper is using multiple methods to characterize this population of organisms. So PCR amplification on the left, essentially. PCR product would be the library insert, or gDNA as your potential insert. One is more targeted. One, you can quantify presence and absence and more or less be sure of the detection level. Shotgun metagenomics, sorry, I keep looking here for the mouse and it's not on the screen. Shotgun metagenomics uses a different sequencing approach. So the confidence in the calls are greater. Generally with Illumina sequencing, the shorter the read, the more confident you can be in the calls. You need a lot more data, right? You've community data with potential lots of variation. You're gonna need more data, whereas if you were just to be looking for short fragments of an organism. And then, yeah, the cost difference is here. The cost here really comes down to the library preparation. Outside of that, there's no cost difference really across pipelines except the library preparation and the sequencer you're putting it on. All right, so let's just briefly talk about what metatranscriptome sequencing is. So it refers to the complete set of RNA transcripts in the microbial community. It reflects the active genes of the microbial community, provides insight into the gene expression of microbial functions, gives us an understanding of the behavior, if they have behavior, if we can give bacteria that phrase, of the organisms, and it shows how they're responding to the environments they're in. So this is the generalized workflow here. So we have total community RNA, and we're going all the way down. This is probably the most important step. I have a slide dedicated to this, so we'll talk about that. Here is the 16S analysis, and then we can further get more of a community understanding at a wider range with metatranscriptome. Okay, so extractions with RNA. I mentioned earlier, RNA is super tricky. It's hard to work with. You can think you're doing everything right, and the next thing you know, you end up with just water out of the column. So your sample source is really one of the biggest hurdles here. For any of those of us lucky enough to just work in water, in my opinion, one of the easiest systems to work in. You know, feces is notoriously hard to extract RNA from. You also run into host contamination issues that Kyle mentioned earlier. Are we getting a lot of information from our host? If we are, it's going to throw off our library and give us a data skew, which we do not want. So are you taking single points of extraction, or are you doing a cross section across your location? Has it been pre-processed before? So I guess kimchi doesn't have that much processing in it, but human intervention in a sample, you should take into consideration the potential inputs that may have gone into it. So if we're dealing with food, could a food worker at a factory line have introduced a contaminant? You should be thinking about that as an, oh, well, this might not be a true representation of my sample. Ways to pre-filter or post-filter, depending on the reference genomes you might have. Also, just money in general. These types of extractions are costly. So rRNA removal of the metatranscriptome. So please forgive me, this is a mammalian cell composition, not a microbe, but it serves the sake for a good example. So the reason why we want to remove rRNA from our microbial community sample is that it makes up a vast majority of the components of the sample, right? So by size here, this is also an issue, is can it be a physical hindrance to the molecular machinery? Usually scientists are looking at a large portion here. And so rRNAs, while still like the second largest, can have a large impact. tRNAs almost never get picked up into Illumina library constructs. You have to do some special stuff. They're really short. So it's not common to be picked up. So if we just remove tRNA, then we're going to have a whole bunch of RNA left over. We don't need this information. RNA, like I said, is conserved. We can just look in a database. So removing this allows us to zoom in on the portion of interest, provide greater confidence in it. Provide greater confidence in it and not waste our money sequencing things that do not need to be sequenced. So here's the generalized library workflow, right? Total RNA, we're going to remove the ribosomal RNA. Fragment, just like all library productions, reverse transcription, second strand cDNA synthesis, and repair and tailing, daploligation, size selection. This can be very important. So ideally, you'll want to ensure that your fragmentation is uniform. This can be species dependent, depending on the enzymes that are being used for theory. So if you have incorporation of a fragment that's 150 base pairs versus a fragment that is 75 base pairs, you will see a dramatic skew towards your 75 base pair, just because, as mentioned earlier, the clonal amplification on the machines themselves. So balanced libraries, always a good idea. Here's the transcriptome pipeline. So as always, with everything that we do, We give out raw data, quality control before it goes to the client. Client approves, and then everything else is given up until this point. And then these are some of our bioinformatic packages. So here on the left, we're just talking about functional annotation. And then we have some enrichments here. So these are pathways, repositories of genetic information down here. So we're going to query against what's known publicly. So the results of the study through the use of amplification, metatranscriptomics, and shotgun metagenomics showed the species, the archaeal species, was the one involved in the degradation. And it showed, as Kyle mentioned, we were able to identify 2 unique pathways that were previously unknown in this type of archaeal environment. Okay, so laptops running low, so significance of NGS data, right? So what's the importance of having this? Yeah, it's over here. What's the importance of having this after this study? You know, it helps identify and provide reassurance that using multiple methods can help provide information in gaps that may have been missed. So yeah, the main conclusions of the study: non-synaptotrophic methanogenic hydrocarbons, gray degradation is, unique to this species and provides us a good insight of what is going on in the system. All right, so if we were to choose, if you're looking and you don't know which way to go, we have a couple pipelines here. So we have resequencing, we have shotgun metagenomics, we have Amplicon metagenomic sequencing. So it depends on our organism, it depends on our end goal, and it depends on the environment we're coming from. Some environmental hindrances will maybe exclude you entirely from whole genome sequencing. Maybe you want to start with Amplicon, identify presence, absence, then go further into the genetic information.